All right. Choosing a research project design. So one thing that I think was eye-opening for me is not everything has to be a big randomized controlled trial. And it can take years to get that done. I think to have most residents kind of expect them to come in and be, spearheading a randomized controlled trial as their first trial is quite unrealistic. And, the baseline goal of scholarly activity is to get you exposed to the process. And the process didn't make a lot of sense to me for a long time. And so that's kind of where I want to go with this, is kind of define these, because you can't just choose a hypothesis and say, I'm going to design it this way. Your hypothesis is really going to determine how your study is designed. Okay. So this is one way to organize it. So we're talking about clinical research at this point, not quality improvement. So we'll talk about that probably at a later date. You can have clinical trials that are observational or non-experimental, meaning you're observing things. You're not putting any intervention on the case subjects at all versus interventional. So all your observational studies include things like case reports, case series, cross-sectional studies, case controls, cohorts, meta-analysis, and systematic reviews. And then your interventional trials are either uncontrolled trials or controlled. They can be randomized and non-randomized. All right. Similarly, you can't just choose a study design and decide you're going to do it retrospectively or prospectively. Only a few of them can do it that way. So cross-section, we'll talk about each of these a little bit more in depth, but cross-sectional studies essentially are just looking at the present, a cross-section in time. So you can't design a cross-sectional study that's retrospective. That makes no sense. Okay. Case control studies are only retrospective, so you can't design a prospective case control study. Cohorts are really, as far as observational studies, the only ones that you can do both retrospective or prospective. So hopefully it makes a little bit more sense to you that it's not just like you can't just design what you want. These are pretty algorithmic in what they do and what they tell you as well. All right, case reports. Measured outcomes, you're really just describing what happened to a patient, and so there's really no measured outcomes from these. They describe clinical events of one or more events in narrative form. There's really a kind of a low-hanging fruit for residents, but don't rely on it. I would say if you're going to do it, but have something else going on in the background, because it may be quite difficult. I have cases that I think are quite good that nobody will publish because it's just not novel enough. It's a rare, super rare case, but they've already described rare cases. I mean, people don't want to read 10 case reports on a rare case. They want something else that's going to add to the literature. So case reports do not need IRB approval at our institution. The Office of Research and Innovation still wants to read them before you publish them. And that's actually quite helpful. They're not like the IRB. They're pretty quick to respond. It's to the point now where I don't even put a consult in. I just email them and to look over a case report, and within like 24 hours they send me back an edited version with spelling errors, or like, hey, have you thought about changing this to this? So they actually provide some super useful insight, and they'll actually review. They're almost like the first submission of a paper. They'll also review the journal that you want to target, their guidelines, and be like, hey, this format has to be. It may save you a month for them to do that. So that's super helpful. All right, case series. The definition of case series is super vague and varies widely. So according to the Dictionary of Epidemiology, a case series is defined as a collection of patients with common characteristics used to describe some clinical, pathophysiological, or operational aspects of a disease, treatment, or diagnostic procedure. They can be retrospective or prospective. They may be consecutive or non-consecutive, depending on whether all cases presenting to the reporting author over a period were included, or if you only select a portion of patients. So this is not nearly as structured, why it also doesn't have as much clinical backing as a randomized controlled trial. You're essentially taking a series of patients that you see with similar diseases and identifying them in some way. You're really describing them. There's not any specific outcomes you're necessarily measuring from them except for what already happened. You're kind of describing more as the way that I think of it is multiple case reports put together. The minimum number for it to be a case series isn't well defined. Some authors define that it should really contain no less than 4 patients and no more than 10, but I've seen other scholarly reports suggesting up to 100. So the actual definition of how many patients in a case, how many patients it takes to make up a case series varies wildly. All right, cross-sectional studies. So this was kind of my aha moment, I think, for this. So cross-sectional studies can't tell you really anything else except for prevalence and odds ratios. Those are the only things that you can get out of a cross-sectional study. So you can't design a cross-sectional study and think you're going to get like case fatality rates and stuff like that. It's just not going to happen. So if you're just interested in finding prevalence, like where's Jace? Not here. Oh, so like the bacterial vaginosis, that's really a cross-sectional study, you know, so we're just trying to find out the prevalence of two diseases. And so I don't know why the IRB said it was really a quality improvement. It's definitely cross-sectional, but whatever. So these are rather quick to do. They don't really need a follow-up period. You're just looking at a snapshot in time, and that time doesn't have to be like one day of chart review. You can do that over like a year or so. But essentially, it's one snapshot in time. Not looking back into the past. You're not following them into the future. They cannot tell you which came first, the exposure of the disease. They essentially just tell you the prevalence and perhaps the odds of that. And they can be descriptive or analytical. So don't get bogged down on this slide. I just want you to show you. Do you guys remember this from like basic biology, Punnett squares? So this is all you're doing in a cross-sectional study. You're taking essentially 4 groups. You're calculating, or you're putting the patient's observed outcomes in the different groups, and then you're determining a prevalence or odds ratio. I'm going to put up a scary slide. So this is to show you really don't need a statistician for cross-sectional studies. This is pretty easy math. To get like power and to get confidence intervals, you may want to involve them, but the whole point I want to get across this is cross-sectional studies are pretty easy to do. And I would consider in the grand realm of doing actual research, going through the IRB is a good starting point for residents to kind of do if you find a topic that's important for this. This is also for odds ratio, again, simple math. All right, case control studies. This is where it starts to get a little bit more in-depth and difficult. So this compares 2 groups of people, the cases and the controls, and it's only retrospective. You can't do a prospective case control study. It cannot tell you anything about incidence or prevalence of a disease. It only tells you the odds ratio. Cohort studies. So cohort is Latin for groups. It's essentially what they used to describe military battalions were different cohorts, and so that's how this is so named. So it essentially follows 2 groups or cohorts of similar outcomes, and so this can measure a lot of things. So this isn't just prevalence or odds ratio. This can tell you just about anything. So you can design this study to get incidence, mortality rates, case fatality. You can do odds ratio and prevalence, relative risk difference, hazard ratios, all that stuff. So this can be prospective or retrospective. Both cohorts should include similar patients at risk of developing a disease. Probably the most famous example of this is the Framingham heart study. You guys remember that? I think they have multiple arms in that, but kind of the biggest thing was statins and heart disease. All right, meta-analysis and systematic reviews. I put these together because you can't really do a meta-analysis without doing a systematic review. Now, systematic reviews can be rapid or comprehensive. Comprehensive reviews should fall in line with guidelines from either Cochrane or Campbell. They're kind of the 2 standards. And so they use the Prisma flow sheet, which you see on the right. Essentially, in 2009, there was an international group of experienced authors and methodologists that developed Prisma, which stands for Preferred Reporting Items for Systematic Reviews and Meta-Analyses. And this is a 27-item checklist and a 4-phase flow diagram. And if you guys have all read through this, we just did this at our last journal club, but if you read through a meta-analysis, you'll see that they have this chart on there, right? So that's how they kind of determine what articles they're going to include. And so it's a flow sheet that you go through. So that's where they get this chart from. is Prisma. So you essentially put it in there, you define it in your methodology which ones you're going to include or not. That's how you specify that they make the cut. And then the MECIR standards is methodological expectations of Cochrane intervention reviews. And so this is a 4-part document essentially that just gives guidelines on how to do this. So you can find that on the Cochrane website if you're so inclined to do a meta-analysis. There's different statistical software that you'll likely need, and because this can tell you just about anything as well. Mendeley, Ryan, EndNote, Zotero, and RefWorks are all software that can actually calculate statistics for you too. Again, it's nice through Michigan State that we have a statistician that can work with us. You can crunch some of your own numbers in this stuff by using that software, and some of it is free. This is that Prisma checklist. We're not going to go through it, but there is, again, 27 points checklist essentially that you have to do. And that's if you want to do a complete review. So certainly a systematic review that follows this guideline, follows a Cochrane guideline, is certainly going to be held to a higher standard than a review that doesn't follow that at all, because you have no idea if the author is including everything or not. All right, and then we're going to briefly touch on experimental studies. So this is essentially the other side of that flow sheet. This is anything that you're going to do that's actually going to introduce some sort of intervention or test on a patient. So these can be uncontrolled or controlled. Control studies can be further subcategorized as randomized. Cohort allocation is based on a random chance, or non-randomized, patients intentionally placed in different treatment arms. Clinical trials can also be classified by their purpose. Prevention trials assess ways to prevent disease using medicine, vaccines, lifestyle changes, etc. Screening trials assess the best way to detect diseases or health conditions. Diagnostic trials, treatment trials, and then quality of life trials. There's 4 phases in interventional trials. Not necessarily all interventional trials will do 4 phases. This is typically for new things, such as the mRNA vaccines for COVID. So that's the big thing. Everyone's saying like they don't follow the trials. They published, and they didn't even finish phase 4 data. They're not supposed to. So phase 1 is typically the first in-human trial. So this is after animal testing if they do that. It's usually just on a small group of people. They're not specifically trying to find out like the effective dose of medications or whatnot. They're trying to see, if I give you this medication, are you going to live, and what kind of adverse effects are you going to have? So essentially, they're mainly just looking at safety, and because they're just looking at safety, they don't want to blind that. So they want to know who's getting it, so they give it to everybody. So this is an uncontrolled trial. Phase 2 trial is typically uncontrolled as well. They usually test it. Okay, so the first small group did okay. Let's test it on a larger group. They may start to test efficacy of dosing at this time, and then they're again looking at safety profiles. Phase 3, this is where you typically get your randomized controlled trial. So this is where they release the data on the mRNA vaccine. They did 30,000 patients, and they said they actually blinded them. So they put them in randomized controlled groups. They again assess safety, but they're mostly looking at efficacy at this point too. And then phase 4 is after it's kind of gone through that, and they open it up to the general population. The medication's on the market, it's out there, and they're doing long-term data on this. So they're looking to see, years down the road if there's birth effects and stuff like that from vaccines or whatnot. So just an overview of the phases. Hopefully it makes sense. All right. Uncontrolled trials. A control group is made up of a group of patients that do not receive the investigational drug, but instead take standard treatment, approved for the disease on which the study is being carried out, or takes a placebo, which is a simulation of the interventional drug. An uncontrolled trial is rightfully named because it contains no control group. So again, the example being kind of like the early phase 1, phase 2 trials, we really just kind of want to know the effects of this drug on patients, not necessarily the efficacy of different dosing and whatnot. That'd be the good design for an uncontrolled trial. If it's anything outside of that, it's not considered super high quality because it's super prone to bias, because you're just putting patients in this. There's no control to it. There's no blinding. There's nothing. Non-randomized controlled trials. And the other thing with all clinical research, it's not so much, hey, I can determine prevalence or odds ratio. You're setting what you're looking for. So specifically, you're defining a primary and secondary objective from these trials, and that's going to be kind of your end goal. And the statistics behind that are essentially what you define for that. So you can do just about anything with that. Non-randomized trials are controlled but not randomized. Examples include placebo-controlled, no treatment-controlled, so one group might get treatment and one group just gets no treatment at all, not even a placebo. Active treatments, control with dose comparators, historical controls, which is essentially information from controls or from patients treated at an earlier time and or in different settings. That's what historical control is. And then kind of the holy grail of clinical research, the randomized controlled trial. Again, you set a primary, secondary outcomes or multiple secondary outcomes on what you want to measure. Subjects are randomly assigned into an experimental group or a control group. As the study is conducted, the only expectation difference between the control and experimental group is the outcome variable being studied. Randomized controlled trials are suitable both for preclinical and clinical research. Should be sufficient uncertainty about the utility of an intervention. That's what is referred to as the equipoise. So if you've ever heard of controlled trials being stopped early for equipoise, that's what they mean essentially. Observational studies are prone to bias. Bias is the tendency of any factor associated with the design, implementation, evaluation, and interpretation of the results of a study to make the estimate of the effect of a treatment or intervention deviate from its true value. If two or more groups are being compared in an observational study, there are often systematic differences between the groups, so much so that the outcome of the group may be different because of these differences rather than actual exposure to the intervention. This is known as confounding. Randomizing is the best way to minimize confounding. So this is kind of your highest quality of evidence in clinical research. So we took that kind of in order of lowest quality to highest quality, which also kind of goes in order of easiest to most difficult to complete. All of them are considered clinical research, right down to a case report. So I guess my advice is you don't have to be the one to come up with the next randomized controlled trial. That can take years to do. I think it's important to have that in our program though too. So if there's residents that want to tackle that on, I also think that you shouldn't feel down or bad about yourself if you don't get it done in a 2 to 3 year time period. So there can be other people that come on and kind of carry the torch along if you can't finish it. So I think being involved in maybe more than one study is helpful too. If you get a couple residents, on board doing a study, and kind of teamwork makes the dream work, we can get a lot more of these kind of accomplished.